first nationally recognized standards for cannabis and hemp products with the American Herbal Products Association, and he is an author of the American Herbal Pharmacopoeia's Cannabis Monograph. Thank you, Jahan. Great to see you. Thank you for setting up the slides there. We look forward to the presentation. Take it away.
All right, thank you. Thank you. Today's presentation is entitled Understanding the Endocannabinoid System and Cannabinoid Receptors. As I was introduced, I have a lot of experience working in academia, policy, and industry. I'm a founding partner at the consulting firm Marcu and Aurora, and serve on multiple expert government, advisory, trade association, and scientific committees, as well as being the editor-in-chief and co-founder of the American Journal of Endocannabinoid Medicine here in the States. I often like to start my presentations with quotes, and this is a quote from one of my colleagues that appeared on the show How to Launch an Industry. The effects of CBD seem to be really strongly conserved across species. We seem to use the same doses in mice, in rats, and humans, which is surprising and interesting, and probably telling us something about CBD's effects. Cornell Laboratories has done extensive studies with CBD in companion animals, and they recommend the same doses of CBD in companion animals for anxiety as we have found work in rats and in humans. And I think today's presentation about some of the fundamentals about the endocannabinoid system may help shed some light on some of these mysterious effects of cannabinoids. So today I'm going to attempt what has never been done before in endocannabinoid science, and that is talk about all these three topics within 30 minutes. So we're going to define the endocannabinoid system, the enzymes, the ligands, the receptor components. We're going to explain in detail cannabinoid receptor structure and function, and end with the potential clinical applications of endocannabinoid system manipulation. Don't get lost in the jargon and the big words if you're not familiar with cannabinoid pharmacology. If it is important, it will be repeated many times. So before we begin and jump into talking about what we know about the endocannabinoid system, some general rules for our journey today. Cannabis and medical cannabis and cannabinoid medicine is based on compassion. However, you need more than that. You need scientific research, and you need research where the results are trustworthy. But we also have to balance that out with the real world, where human beings are notoriously underfunded for cannabis and endocannabinoid research and have very short deadlines to turn around their data and publish it. So always wonder about how well these studies are controlled, whether or not they're reproducible, if they are using a product, if this is the provenance of the product known. Always think about what are the applications of it and how people are filtering and what lenses they're using to either gather the data or look at the data or pull out their conclusions. And when you're doing your own analyses, always think about, are you using the all available data that's out there, or are you using the highest quality of data that's available? And with that, we'll begin our dive into the endocannabinoid system. So this system, the ECS system, was discovered with the help of plant compounds, notably those found in the cannabis plant. However, many other plants have been shown to produce what we might call cannabinoids. THC and CBD, for example, are only found on the cannabis sativa, aka hemp, aka marijuana plant. The endocannabinoid system, the ECS, consists of endocannabinoids, endogenous or internally produced compounds, receptors for those compounds, and proteins that are important for their synthesis and breakdown. What does this system do? A phrase that was coined a long time ago is this system helps us to eat, sleep, relax, forget, and protect. Many of us may be familiar with the effects of cannabis on stimulating appetite. Stimulating appetite is not something new that cannabis introduced into human biology. It's been there a long time, and the system that helps regulate that is the ECS. It helps us to sleep, it helps us to relax, such as having anti-inflammatory properties. It helps us to forget, and it helps protect our healthy cells from damage. I believe that forgetting is probably one of the most important aspects of the endocannabinoid system. If I remember every face that I see on the subway here in New York, my head would explode. You need a system in your body that helps you get rid of useless information and keep what is important. Or, for example, allow you to focus on giving a PowerPoint presentation and not whether or not you remembered to feed your cat. When things go wrong in this system, or there are mutations or deficiencies, that has been coined the clinical endocannabinoid deficiency by Ethan Russo, and we'll discuss a little bit of that at the end. So, we have a bit of a family here in the ECS. There are two main receptors that have been identified, the CB1 receptor and the CB2 receptor, both identified by researchers in the United States, and they're members of the G protein coupled receptor family. Generally, when we talk about the CB1 receptor, it results in the inhibition of neurotransmitter release due to hyperpolarization of membrane potential in the pre- or postsynaptic neuron. If you can memorize that and say it at a cocktail party full of medical doctors, they will all be very impressed. CB2 receptors are primarily associated with cells derived from the bone marrow lineage. That sounds important. It is. Those are where your immune cells come from. That's where your cells come from to build your bone, and more on that later. However, there have been orphan receptors that may be cannabinoid receptors and contribute to the activity of this system and of cannabinoids. What are orphan receptors? Well, they're receptors that exist in the human body, but we do not know what ligands, what natural endogenous compounds made by the human body stimulate those receptors. Other things like ion channels that we will discuss briefly may play a role. So the endocannabinoid system has some interesting activities, and for the pharmacologists in the audience, this might be of interest. So CB1 receptor has a number of activities that we will show with this model of a politician's brain. And so this is an accurate model of a politician's brain because there are only two receptors that are functioning, or two brain cells that are functioning. And so basically when CB1 receptors on a brain cell or neuron are stimulated, you get inhibition of certain types of calcium channels. You get the stimulation of potassium channels. You get activation of MAP kinase, and you inhibit adenylate cyclase, which are all important things that lead to what? Inhibition of neurotransmitter release, such as glutamate, which can become important during injuries or excessive activation, such as excitotoxicity. CB2 receptors, similarly, not shown here, can inhibit adenylate cyclase as well, and their activity also leads to the activation of MAP kinase. So, how does this all fit together? Well, when you have endocannabinoids stimulating a cannabinoid receptor, such as a CB1 or CB2 receptor on a cell, it leads to a lot of interesting things happening, not just at the level of the receptor, but at the level of the cell, turning on and off different ion channels that help the cell go about its business. So where are these cannabinoid receptors? Where do things like THC go in the brain? They go to the basal ganglia, they go to the hippocampus, they go to the cerebellum. And some people think that there is some sort of central region, like a Snoop Dogg of the brain, where the cannabinoids go. That is not true. CB1 receptors, the type 1 receptors that we usually associate with the euphoric effects of THC, with the appetite enhancing effects, with certain other effects of cannabinoids, is found just about everywhere. Widespread, high level of expression, and its distribution parallels its known pharmacology. So if it's affecting muscle activity or emotion, we find it in those areas of the brain. We also find it in the nucleus of the solitary tract. That's important. It's for appetite. It helps affect that. The hypothalamus, our motor systems, found in the eye, sympathetic ganglia, and throughout the GI system. It is also found in our immune system, not quite as abundant as its friend, the CB2 receptor, and it's found in a lot of other peripheral sites, your heart, your lungs, your kidneys, your liver, and so on, as well as in the reproductive organs. Now, the type 2 receptor is most abundantly found throughout the cells of the immune system. your monocytes, your natural killer cells, your mast cells, and the level of expression is increased during different pathologies, which is very interesting. So if you receive an injury, such as a head injury or a leg injury, pick an injury, CB2 receptor expression is going to increase to try and control the inflammation and seal off the area. This can be a good thing or a bad thing, depending on what's happening. So there's some very interesting research in this area on cannabinoids and stroke. As well as you can find what's most interesting to me, because I spent a lot of my thesis studying it, is the role of these receptors in bone cells and bone health. And CB2 polymorphisms in humans are associated with more severe age-related osteoporosis. More on this later. But also the mice that have been genetically bred to not have CB2 receptors have Swiss cheese for bones, although this is a little bit controversial because they have a number of other health issues. CB2 receptors are also found on the microglia. You may know them from brain cancers known as gliomas, but they do play a role in the immune system of the brain, and there is some evidence for their activity in brainstem neurons. Cannabinoid receptors are not just found on the surface of cells and the surface of cells throughout the body, but inside of cells as well. And some of our research has shown that you can indeed target CB2 receptors that are in different aspects of the cell. So as we develop new types of drugs and delivery systems, sure, the age-old delivery system of inhaling cannabinoids gets to the cell surface. We have seen animal studies, human studies, where there are injections of THC and other cannabinoids into the bloodstream. And now we're starting to see much more complex formulations that could perhaps target gene expression, target protein folding in the Golgi complex, perhaps target mitochondria, target receptors as they're shuttling back and forth from the cell surface to inside the cell. A very interesting area of research there with how we could perhaps pinpoint and target cannabinoid receptor activity. So, what do we know so far? Well, cannabinoid receptors are basically everywhere. So, when we talk about the cannabinoid receptor structure, typically we talk about these 7 transmembrane helices. So ignore the dots for now. It's just showing some key amino acid differences between CB1 and CB2. So if you're a molecular pharmacologist, you will probably really like this slide. So what we see is the top part of the receptor is outside the cell and the bottom part is inside the cell. Now, the cannabinoid receptors don't exist like these really neat flat packages because the cannabinoid receptors are made up of amino acids which make up proteins. And where do amino acids come from? They come from gene transcription. I know it's a long trip back to grade school biology for some of you, but these amino acids all have a little bit different properties. Some are positively charged, some are negatively charged, some are big and bulky, and they contribute to different properties of the receptors. And some of you who are acutely paying attention may have noticed there are 2 residues in red, and these are very important for stimulation of the receptor by drugs. So if you mutate these to something, say, not positively and negatively charged to hold it together, but more just neutral charged, like an alanine residue, for example, certain aspects of the receptor won't work anymore. So, like I said, we have these amino acids that are not laying flat like a snake or something like that, or a pressed wildflower. They are indeed folded up in a complex 3-dimensional structure. And on the top of your screen, that's where, near that blue ribbon that's poking out, that's where a lovely cannabinoid might enter the receptor for an interaction and convey its signal to the rest of the cell. So how does this work exactly? Well, you have, there's our initial green model of the receptor, the inactive receptor with a beautiful G protein complex. When an agonist, such as anandamide or THC, stimulate the receptor, this G protein complex dissociates and goes about its cellular business, turning on and off things throughout the cell, having some downstream cellular responses as we showed in earlier slides. However, if you have disruptions in this receptor, Maybe certain agonists, certain ligands like anandamide or THC may not bind or stimulate the receptor anymore, thus altering the signal that is conveyed to the cell. So, what sort of tools do we have to study this? Well, as I mentioned before, cannabinoid receptors are just proteins. Proteins are made of amino acids, and amino acids have different properties, like the difference between bricks and beams and concrete. Um, some are important for binding, that's the key and lock. Does it fit in the receptor? Some are important for activation, such as does this lead to a conformational change? And so other amino acids obviously play a structural role in maintaining the integrity of the receptor. And so what we're looking at here is one of our computer simulation models that has been run based on mutational analysis data to help develop new cannabinoids and new understandings of the cannabinoid receptor. And this is sort of if THC or anandamide was parachuting onto the top of a building, it's sort of the last thing an endocannabinoid or phytocannabinoid might see before it stimulates the receptor, diving into where those two green amino acids are indicated, thus causing that salt bridge to break apart and the receptor to move and convey a signal. So you've heard me talk about a little bit of an alphabet soup about compounds. We have a lot of different ones. We have bicyclic cannabinoids. Those are the ones with 2 rings. Tricyclic, 3 rings. Glad somebody said that. You also have indoles, which are amino alkyl indoles, WIN55212, and some of the more infamous compounds like the JWH series and synthetic cannabinoids tend to be these indoles. Funny thing about indole derivatives, they do not have the same binding and activation requirements as the tricyclic and bicyclic phytocannabinoids that we mostly know and appreciate. So if you mutate, say this important aspartate residue, 2.63, you can get rid of anandamides binding to the receptor, but the synthetic indole derivatives will still have activity. So a lot of these indole derivatives that are sort of demonized out in the media were actually created as therapeutic agents to stimulate cannabinoid receptors in the event that there was either your typical THC marinol didn't work or a receptor mutation that didn't allow the normal activity of the receptor. And we're going to talk a little bit about what happens when the endocannabinoid system goes awry in humans at the end of this presentation. We also have the fatty acids, anandamide and 2-AG, as well as antagonist, inverse agonist, and we'll get into a little bit more of these definitions very quickly. And you have assays, I said, research experiments to study these that are very well established. So, there are a lot of different types of receptor activities, and I'm happy to share this presentation if you'd like to reach out to me, or this research study that was important for establishing the nomenclature of cannabinoid receptors for IUPAC. So we have orthosteric and allosteric binding. Orthosteric is the primary binding site. Allosteric is something in Latin that just means that other site. We have agonists and antagonists. They're the things that directly stimulate the receptor, things that sort of block the receptor. And we also have allosteric modulators. We have NAMs and PAMs, some prime it for activity, some prime it make it more difficult for activity. And just to make things just a little more complicated, these receptors, again, don't exist in a flat plane. They exist in folded up charged bundles, which can also form associations with other receptors. And for some good bedtime reading, you can look up heterodimers of cannabinoid and opioid receptors. Fascinating field of research there. So as I mentioned before, and you're all very excited to hear about, what happens when receptors start to mutate in the wild, in human populations, in animal populations, and out there in the mammalian world? Well, we see mental health issues. We see substance abuse issues. We see issues with memory. We see issues with eating disorders, which make a lot of sense. In a study of anorexia bulimia patients, it was shown that a population of people suffering from this affliction did indeed have polymorphisms or differences in their CB1 receptor, the one that's important for appetite in a population. Fascinating stuff. NCB2 receptors have been shown in at least 3 distinct human populations to be related to more severe age-related osteoporosis. This was shown in postmenopausal women in France and Japan, as well as in a community of people living along the Volga River in Eastern Europe. So, cannabinoid receptors and the genetics thereof are not just some quiet hobby for nerds to engage in an ivory tower. These are real studies that have clinical implications, and looking at the way that genes can affect people's response to THC or CBD or other cannabinoids is currently ongoing. Check out clinicaltrials.gov to see some more interesting pharmacogenetics or pharmacogenomics trials currently underway. So great, this is all fascinating, but how do you use it? So I'm going to talk about one case to wrap up here. So we have a fairly curious endocannabinoid case in pain. So what if I told you there was a person out there where morphine did not relieve their pain, but they also didn't need pain medication even after surgery? Opioids had no effect, had a high threshold for pain. Could the endocannabinoid system explain it all? So here we have a case report of a 66-year-old female, no pain medication required. History of painless injuries, stubbing toes, cutting fingers, doesn't bother her so much. Turns out that she had high levels of 3 of the main endocannabinoids. It turned out she had a Fa-out single mutation. So this basically means that the enzyme responsible for breaking down the endocannabinoids had severely decreased activity and expression. It just was not being translated very much by the nucleus and sent out into the body to break down endocannabinoids. And so what this meant was she had a higher level of cannabinoid receptor activity, most likely leading to a lot of, pain desensitization. So, the mutants walk among us, and so far we can tell Fa pseudogene is potentially one of those mutants. So what are some of the implications of having an ECS that, say, is not functioning to what we might call national standards? Well, no endocannabinoid system, no Tylenol. One of the interesting things in a study was shown that a Fa inhibitor that goes into the brain actually abolished the pain-relieving effects of paracetamol or Tylenol. And so if you Tylenol basically could be considered one of the first over-the-counter endocannabinoid pain drugs. So again, the endocannabinoid system, just to wrap up, is very important for the effects of Tylenol once it has been metabolized. So there are some variables, as with everything, in terms of what we can say. So we've talked about the genetic components, right? Certain genes are altered, it changes your response. We didn't even talk about liver enzymes and drug metabolism. We'll save that for the sequel. But dosing and administration can play a role. How are people taking these drugs, and how many different, what amounts, and are they combining administration forms? Inhalation is a very popular delivery form for cannabinoids if you have not been paying attention, but people inhale things very differently in terms of amounts and intensities and for lengths of time. You also have disease conditions. So if, say, you have skin injury, twisted ankle, some sort of cardiovascular event, your CB2 receptors will elevate. Lifestyle and diet also do play a significant role in the health of your endocannabinoid system. The ECS is not a closed loop system. You need to get things from your diet to fuel it. You need omega-3s and omega-6s, and indeed, without those, the system just can't function properly. So thank you so much for your time and attention. At my firm, Marcu and Aurora, please reach out to us. We help with corporate advisement, investor due diligence, so we do custom training and education for people all over the world. I myself am also teach at universities. Science communication is something we train a lot of C-level people in. We do experimental design and review and facilitate relationships with regulators and academia and other industry partners. Be sure to check out the show, How to Launch an Industry, fascinating science-based show looking at cannabinoids and psychedelics and other research topics. Thank you so much, and happy to take a few questions if there's time.
Much, Jen. Yeah, if anyone would like to ask any questions, please do ask away. We have a few minutes spare as we go into this next section. One thing that you were kind of consistent with throughout there is the regulatory issues that we're going to come on to. Partly there's a part of the next panel, but definitely later on as well. And obviously, you've worked crossing from like the academics over into the industry. Are there any kind of key unifying regulatory issues that affect the academic, the industrial, and those providing the services to this area?
Sure, I think it's always an issue if you're in whether private R&D or in a research setting that you have regulations that allow you to access your research tool, and most specifically the cannabinoids you might use to explore receptor function or clinical effects. And it's not always so clear cut at universities like it might be in the venture capital space. What people can do with CBD, for example, in the United States, if you just have a unlicensed CBD company going wild, is very different from what you can do in a university setting. Even though the university setting perhaps has more resources, more data throughput, perhaps a higher impact with the research that comes out of it. And so you have to balance those things and sort of weave in between them and take the best of both worlds. But I think the key factor is that whether it's academia, whether it's industry, whether it's a policy or regulatory office, they cannot survive without each other. They all need the experts from each other to sort of work together to come up with the best environment.
Perfect. Mauro had a question for you. He's written it on the side, but you also requested to come on screen, so I thought, why not bring him on screen so you can ask it away. But do you want to ask your question, Mauro?
Yeah, so thanks again. It was a nice presentation. I was curious to know if you ever interrogated cholesterol binding to CB1 that recently in the Cell paper by Hua was highlighted as a very interesting point of action. And cholesterol has apparently more than one sequences that are specific in CB1 for its binding, like crack and others. Have you ever interrogated something like that?
Not directly. One of the ways that cholesterol is important is when you are doing the binding assays, but to be perfectly honest, we were never 100% sure what its role was, but it is there when you are making the cell preparations to do the ligand binding. But I think it's a fascinating area, and if cholesterol can be some sort of allosteric modulator at the receptor, I think that there's a very interesting implications there for diet and consuming cannabinoids and how their effect can be improved or inhibited. But thank you for bringing up that point. I'm going to check out that paper and incorporate it to my presentation in the future.
You will find a few.
Thank you.
Thank you very much, Mauro. Great. That concludes that. Thank you for your presentation, Jahan. We're just going to invite up the panelists for the next panel discussion on the agenda, which is the panel discussion comparing whole plant and synthetic cannabinoids and their potential in pharmaceuticals, and that is going to include Mike Woodenberg, Aurelia Dapau, Tamas Zdenkeni, and also Michael Swardegren as well. If you could all hit the share audio video button in the top right corner of your screen, and we